Hello, welcome to the Rocky Mountain MS Center Spring Summit. As you know, I'm the medical director and professor of neurology at the University of Colorado. I'm sorry we can't do this in person, but hopefully you will find some of the information provided by myself and my colleagues useful today. So I'm going to be talking about the concept of brain health in general for all of us, as well as special issues related to patients with MS. And our goal here is to really empower you in order to help you make the best decisions you can about your care and your life. Okay, before I do that, I'd like to just briefly introduce you to the Rocky Mountain MS Center and the Rocky Mountain MS Clinic. The Rocky Mountain MS Center is a community-based nonprofit organization established over 40 years ago. It's been involved in patient care, research, and education through all that time. In 2008, I returned to Colorado and joined the Rocky Mountain MS Center as a medical director. At the same time, joined the Colorado University School of Medicine, where we established the Rocky Mountain MS Clinic. And that clinic now has 7 neurologists, 3 advanced practitioners, and about 10,000 patient visits per year with roughly 4,000 distinct patients. Not all of them are MS. We also deal with other inflammatory diseases of the brain, including neuromyelitis optica, autoimmune encephalitis, neurosarcoid, and others. We have a very active clinical research program. And our team is somewhat unique in the world in that we try to decrease practice variation among ourselves by discussing any differences in the way that we're treating a patient, and then use the data that's been published or generate our own data to allow us to make the best decision and optimize patient care. This relationship with the Rocky Mountain MS Center and the University of Colorado Hospital and the University of Colorado School of Medicine has been very productive, and the Rocky Mountain MS Center has supported a great deal of our research over the years. So the topics for today are going to be focused on, initially on talking about the concept of brain health and issues in MS that may impact this. And then I would like to switch gears and talk about a new biology of MS that is emerging that's quite different than the traditional view of the immunology of MS. And the reason for this is to lay a foundation for the third talk, part of the talk, which is to talk about how astrocytes are key players both in the process of injury in the central nervous system, but also key opportunities for repair and neuroprotection in MS. And then briefly close on biomarkers and explain briefly how these new biomarkers that we are studying are going to help us as we continue to understand this biology of MS and improve treatments for patients with MS. So first thing, brain volume and neurological reserve. Brain volume is a reflection of how many nerve cells you have in your brain. And neurological reserve is just the concept that the brain has extra capacity and it can compensate for a certain amount of injury. And I'll explain why that's important. Now, neurological reserve can buffer for subclinical injury to the brain, such as a minor closed head injury that does cause some injury to the brain, but doesn't result in any symptoms. But it also is related to how much recovery function you can get after a significant brain injury that does cause symptoms. And so building and maintaining neurological reserve is very important to help the brain compensate for the normal events that happen in life. To talk about that, we need to have some nomenclature that we all share. In this cartoon of the different cells that make up the brain are basically 4 different types. So neurons, which are the whole point of the brain. Neurons are the little computers that are sensing information and processing it and allow us then to behave and respond accordingly. All the other cells in the brain are there to support neurons. The oligodendrocytes are the cells that make the myelin. And the myelin is the fatty sheath around the nerve fibers that helps the nerve fibers conduct signals at a faster rate. Traditional thinking about MS has focused on oligodendrocytes as the primary target for the immune attack in multiple sclerosis. But I'm going to share some data with you to suggest that's probably not the case. and that rather it's the third component, the astrocytes, that actually may be the first target in MS. And I'll explain how that impacts the rest of the functioning. Microglia are just part of the immune system and are there to help protect the brain. So the key players for us today are the neurons, which are lost in MS, the oligodendrocytes, which are also lost in MS, and then the astrocytes and the roles that they play in the loss of those two cell types. So let's talk about just the natural history of the brain here for a second. We're born with pretty much all of the neurons we're ever going to have. And yet the brain continues to expand in size from birth to about age 20. And that expansion in brain size is due to proliferation of those other cell types, the oligodendrocytes and the astrocytes in particular, and also expansion of the neuronal synaptic network, the number of connections between your nerve cells. So that network of connections expands as we grow and develop new skills. Until about age 20. And about age 20, our brains reach maximum size, and they're pretty stable until we reach our 30s. And then in all of us, we begin to lose nerve cells, which is demonstrated by a slow decrease in brain volume, and I'll come back to that. So this loss of brain volume that occurs beginning in the 30s and 40s is important because it is potentially going to cause us to lose some neurological function, particularly late in life. So again, neurological reserve, trying to expand and enhance it, is important just to compensate for this normal effect of aging. Now, how do you build neurological reserve? I'll go over that in just a second. So first thing, talking about brain volume changes in multiple sclerosis. In this study done by our colleagues in Southern California, they looked at multiple sclerosis and then corrected the amount of brain volume loss for just normal brain volume loss that occurs as normal part of aging. And what you can see here in red is the normal loss of brain volume at age 30, 40, 50, and 60. And this panel up here is looking at whole brain volumes. And what you can see is that initially at age 30, almost all the brain volume loss is really related mostly to MS. But as we age, particularly up in our 60s, the majority of the brain volume loss is now due to aging. This has an implication that if you're trying to prevent brain volume loss due to MS, you want to treat back here, because once you get up here, you can only treat a very small part of the brain volume loss, and therefore you have a relatively small impact on the outcomes. This is also reflected in subunits in the brain, the thalamus. You can see the same basic pattern. It's different, however, in the caudate and the putamen. We don't know quite why that is. Now, the relevance of this difference in terms of how much brain volume loss is due to aging and how much is due to MS is reflected in studies of outcomes of MS patients. So I'm going to argue that the loss of neurological reserve actually is the cause of the progressive form of the disease. And in this study, down in this panel here, done by our colleagues in England a number of years ago using alemtuzumab, but they showed that they monitored patients for a year, and in the blue is progressive patients, and the red is MS patients, relapsing-remitting MS patients. They treated them here at this point with the alemtuzumab. And what you can see is that 6 months later, the patients that had relapsing-remitting MS actually improved substantially, by 2 full points on an 8-point scale, which is very good. But the progressive patients still were a little bit worse, but not changing as fast as they did the year before. And that was true at month 12, 18, and actually out to month 36. So the relapsing-remitting patients are able to get better, and that's because they still have neurological reserve. But the progressive patients were slowly worsening, and I would argue this worsening here is due to this loss of brain volume, which is due to aging up here. Okay. So neurological reserve in MS is the capacity of the brain to compensate for MS disease, and most lesions that form in the brain due to MS are actually clinically silent. 90 to 95% are not detected by the patient or the physician by exam or history. They're only seen on MRI, but they are causing damage to the brain. The brain is shrinking at an accelerated rate, as I just showed you. The neurological reserve is not just related to cognitive function, it's also related to motor function and sensory function. And because of neurological reserve, the diagnosis of MS is often delayed. Again, if 90 to 95% of lesions are clinically silent, you can easily imagine that a patient may accumulate a fair amount of damage to the brain before we actually have a lesion that causes symptoms that leads them to the healthcare system and a diagnosis. And also that loss of neurological reserve is the cause of progressive disability. But patients can increase neurological reserve, as all of us can, by living an active, healthy lifestyle. And what this means is that we have to adopt an active exercise program, because as you exercise, the neurons that are firing begin to make new connections, and they expand the size of the synaptic network that allows the brain to work. Also, eating a healthy diet to avoid comorbidities such as diabetes or hypertension or other diseases is important, because they also damage the brain, and that increases the disability from multiple sclerosis. Also, being socially and creatively active is really important. All those activities, every time you learn something new, you make new connections in the brain, and that expands the neurological network, and that increases your neurological reserve. So an active, healthy lifestyle and and a healthy diet are key parts of maximizing neurological reserve and therefore brain health over a lifetime. I did want to go a little bit more into other factors for neurological reserve than I have before, and this is a figure from a new paper that we have coming out. So if you look up here at the top of the various inputs that determine how much neurological reserve you have, there are genetic and inborn factors, and they lead to how big your brain is going to be and how much reserve you're going to have if everything goes perfectly. And then you've got neural network function, and this is early life activities. This is where children go to school, play, learn to play musical instruments, read, etc. That expands the neural network, and that increases neurological reserve. But then you're also going to have environmental factors such as growing up in a war zone or being malnourished. That could decrease the amount of neurological reserve you have. Or you could have diseases such as multiple sclerosis, which tends to start in early life, that also decreases the amount of neurological reserve. And these are interacting with other factors that we can measure in terms of what you're doing now, current activities that you're doing to increase neurological reserve versus what you've done in the past. And there are also other genetically determined disease-related characteristics that determine the overall reserve. And neurological reserve isn't evenly distributed throughout the brain. It can be different for different subsystems in the brain. So the motor system will have a certain amount of reserve capacity, cognitive system will have a certain amount of reserve capacity, sensory symptoms, visual symptoms, etc. So it is a complex concept. In this graph here, we're just showing what we see happening in MS. So the top line is just normal aging, happens to all of us. The problem with multiple sclerosis is that the preclinical phase, we lose brain volume. That continues if we don't treat the disease, and they get to a point where they just don't have enough reserve capacity to avoid disability. In this bottom graph, we're just trying to demonstrate the different forms of MS that we identify are really all part of a continuum. So the very earliest form of MS is radiologic isolated syndrome, where a person has an MRI because they had a headache or something, and then we see evidence of MS on that. Again, most MS lesions are subclinical, so this is not an uncommon finding. Clinical isolated syndrome are patients who just had one symptom, and then relapsing MS patients, by definition, have had two or more events that led to neurological symptoms. And these patients then move into secondary progressive disease. Primary progressive disease, though, doesn't have the relapses, so it just goes directly into the progressive phase of the disease. And this appears to be mainly just a matter of luck. We have not been able to identify any real biological differences between patients that develop primary progressive disease and those that go in the relapsing pathway. So let's switch gears now and talk a little bit about new biology of MS. To do that, I'd like to review with you a little bit the traditional view of MS, and that is that it's driven by T cells, which are recognizing molecules that are coming from the myelin basic protein, protein, MOG, etc. And a B cell or other cells work with the T cell to demonstrate or show this molecule to the T cell in a way that it can see it, which then activates it and leads to it releasing a lot of small molecules called cytokines that then lead to a whole cascade of events happening. And again, this whole concept is that it's really coming through this interaction with some antigen coming from the myelin. This is supported strongly by the animal model, which is called experimental autoimmune encephalomyelitis. The problem is we've never been able to find this myelin antigen in humans, nor do we find the kind of T cells that we find in the mouse model in humans in a reliable fashion. What we do find are B cells that are responding to something in the brain, and these cells have now become the real focus for what we're trying to do in terms of improving treatment options. So the first thing to understand how things have changed over the last several years is to understand lymphatics in the brain. Lymphatics are small tubes that we have throughout our body that help drain excess water out of tissues. And in doing so, they also carry various molecules. And these tubes is a vascular system that drains into lymph nodes. And then it's the lymph nodes where lymphocytes and T cells, B cells, and other kind of immune cells are. And they're screening that fluid as it goes through to see if there's anything in it that suggests infection or tissue damage. And if there is, then the lymph nodes begin to respond to that. Up until 2014, it was not believed that there were lymphatics in the brain. And it was very unclear how molecules in the brain might be setting off an autoimmune attack that would lead to MS. But in 2014, a group in the University of Virginia identified that there actually are lymphatics in the membranes around the brain, and these lymphatics are imaged here in this MRI. So the green markings here is where dye that was injected into the bloodstream and then leaked into the brain is now being cleared by those lymphatics as it drains out with the excess cerebrospinal fluid. And these drain into lymph nodes that are at the base of the skull, called the deep cervical lymph node chain. And that's turned out to be very important. So again, we have these lymphatics are in the meninges around the brain, and they're draining out to the deep cervical lymph nodes. And so what's happening is that immune cells move into the brain and may get activated there, but they also then drain out through these lymphatics, and they end up in these deep cervical lymph nodes. Spinal fluid's coming out, and spinal fluid is carrying various proteins and other molecules from the brain. And somewhere in that process, it sets off a cascade of events that leads to production of a type of a B lymphocyte, the cells that make antibodies, that then can cause inflammation in the brain. And so those B cells expand in the lymph node, they then re-enter the circulation, they flow into the bloodstream, and they cross the blood-brain barrier, and there they interact with some molecule or cell that activates them, and then they get T cells and other cells involved, and they bring a whole lot of different parts of the immune system into the brain that causes a focal area of inflammation and ultimately damage. Recent data from John Prinius suggests that these B cells are initially killing the astrocytes that are normally in the brain. In fact, half the volume of the brain is made by astrocytes. And so initially astrocytes are dying, and then they come back from neural stem cells and are repopulated. When they come back, they're now pro-inflammatory, called A1 astrocytes. And these astrocytes are producing toxic molecules, nitric oxide, tumor necrosis factor, and other molecules. And these molecules can damage and kill other cell types, including neurons, oligodendrocytes, and actually destroying the axon as well. So it does not appear right now that the attack on the neuron and oligodendrocytes is directly from the T cell, B cell. It appears that it's an indirect consequence of these T cells, B cells interacting, recognizing some antigen that the B cell is now specific for, and that produces these hormone-like substances, these cytokines, that then activate the astrocytes, in addition to possibly killing them initially. So this whole process is much more complicated than was originally thought, but it does give us additional opportunities to actually improve treatment options for MS patients. Traditionally, we thought just focusing on this T cell, B cell interaction over here was the primary goal to shut off the inflammatory disease in MS. We now think, though, that targeting these cells is actually a therapeutic opportunity, and in fact, one that we've already taken. So of all the drugs that we currently have approved for MS, which there's a lot, only one of them was actually developed based on the mouse model, and that's Tisabri. All the other drugs that were developed were empirically derived. In other words, it was because of some effect that they were thought to have in other diseases that somebody thought might be relevant to MS that caused them to actually be tried in MS. The timiracetate over here was actually an accident in the laboratory. But what I've done here is I've just listed all the drugs. The ones in black are actually the ones that are working at the lymphocyte level, the T cells and B cells, and trying to decrease disease activity. And some of the most effective ones are these monoclonal antibodies like rituximab, ocrelizumab, rituximab, ofatumumab, which are all antibodies that directly target the B cell. And that's actually where our knowledge that B cells are key drivers of the disease came from, was the success of these trials. But there are also drugs that we have, which is Tecfidera, Vumerity, Gilenya, Mayzent, and some new ones coming out, and Copaxone, that actually seem to work primarily through astrocytes. And these drugs penetrate the blood-brain barrier. Actually, they get concentrated in the brain, and they interfere with a certain set of signaling pathways that I'll show you in a second. And so their clinical effect can be demonstrated to be due to their ability to inhibit those pro-inflammatory astrocytes. Whereas Copaxone works by developing a B cell that is anti-inflammatory and gets activated in the brain and turns down this process. So let's talk about astrocytes as a new target for neurological repair. So again, to go back to astrocytes are the support cells. These are cells that make up the bulk of the volume of the brain. They surround all neurons and all oligodendrocytes and provide nutrients to them, take away waste, and provide a lot of other support. So these are really key cells and they're very important. But they also can act like an immune cell. So there's in the nucleus of these astrocytes, there's a signaling pathways that can be affected by various agents, pro-inflammatory agents, molecules from areas of damage, are drugs from gamma IL-6. FGY720 is actually gelinia, and it can impact astrocytes because it binds to a receptor called an S1P receptor. And the pathway that leads to the damage in MS is this pathway called NF-kappa B. And when this pathway is activated, it leads to the astrocytes becoming pro-inflammatory. They produce cytokines, they upregulate adhesion molecules that attract more immune cells into the brain, and they also produce nitric oxide, tumor necrosis factor, and other things. But there's another signaling pathway called STAT3 that if you activate that pathway, it actually blocks the inflammatory phase and causes the astrocytes to reduce inflammation, decrease the infiltration of immune cells and demyelination, and help repair the scar to allow the oligodendrocytes and the neurons to begin to regrow and develop new connections. So right now, there's FGY720 or gelinia that does seem to do, at least inhibiting the NF-kappa B part. We're not quite sure whether it activates the STAT3 pathway. But in fact, there are a number of other molecules now that are available that can do this. This list is a long list. These are the traditional approaches to try to remyelinate. And I'm not going to talk about them very much because they haven't gone very far. But I did want to mention one thing, and that is biotin. Biotin's phase 3 trial was completed earlier this year. It did not show benefit. And so people are taking biotin because they hope that it's going to help repair the nervous system in MS. Unfortunately, it does not seem to have a significant effect. It can interfere with certain blood tests, and so I would recommend probably not taking high-dose biotin. The other drugs I'll talk about before, but I did want to talk about two key ones down here. One's metformin. Metformin was recently studied as a way to recover function in the animal model of MS called EAE by Robin Franklin in London. And the reason that he did that experiment is because metformin is a very commonly prescribed drug for type 2 diabetes. And observing people with diabetes on this drug found that if they had a stroke, their stroke tended to be less severe and they recovered better from it than people not on metformin. And metformin does work through astrocytes. It seems to promote that more neuroprotective astrocyte through the STAT3 signaling pathway. So this is a drug I'm actually very excited about and would like to get into clinical trials right away. Then the other one is iluzanimab. This is a monoclonal antibody that works in the MS plaque by blocking a molecule called repulsive guidance molecule A. And that molecule is overexpressed in the MS plaques and prevents the oligodendrocytes from remyelinating, also prevents neurons from making new sprouts and reconnecting to the neuronal network. So we have already iluzanimab in clinical trials, and metformin will be in clinical trials shortly, hopefully at multiple places. So finally, just to close here, talk just a bit about biomarkers. So biomarkers are things that we can find in bodily fluids that tell us that something's happening in some compartment. In the case of multiple sclerosis, we would very much like to be able to monitor the neurons, the oligodendrocytes, and the astrocytes in terms of injury and activation. But the problem is we've never been able to do that on a regular basis because it required spinal fluid. But now we have technology that's very sensitive, can detect one molecule out of 10 with 15 zeros after it. And that allows us to actually monitor blood samples and looking at markers of injury going on inside the brain. And we've shown in our studies here that GFAP, which is a marker of astrocytes, is elevated in the blood in patients during active disease, as is neurofilament light, which is a protein that comes from the nerve axon, and that these two are actually very good markers of disease activity, and I think are probably more sensitive than even MRI is. I expect they will be in the clinic by the end of this year, early next year, as a regular way to monitor it. But right now, they can be used in early phase studies to study drugs that might be neuroprotective or anti-inflammatory, which would allow us to study far fewer patients and do the study much more rapidly. And also be used to monitor patients who are on therapies to make sure that they're getting full treatment effect and to help us adjust therapies when they're not getting full treatment effect. I think it will also help us with progressive disease and understanding a little bit more about it and how we can treat it more effectively. So all of these things are coming together where we are moving into an era where we will be doing more research on neurological repair and recovery of function, as well as neuroprotection to improve function late in life, not just for MS patients, possibly for other people as well. Thank you very much.